Reproduction is the biological process by which new organisms known as the offsprings are produced by their parents. It is of two types, sexual reproduction and asexual reproduction. Asexual reproduction involves a single parent that produces a genetically identical offspring without the fusion of gametes, whereas sexual reproduction involves two parents of the opposite sex. Let's talk more about asexual reproduction. Asexual reproduction is the production of new individuals from a single parent, commonly observed in single-celled organisms. This process does not involve the fusion of gametes. Instead, a single parent organism divides into two or more daughter cells. The offspring produced are genetically and physically identical to the parent, and they are known as clones. Features of asexual reproduction. No formation of gametes or fertilization takes place. In asexual reproduction, the process does not involve the production of gametes like sperm and egg cells or their fusion, means fertilization. Instead, the offsprings are produced directly from the cells of the parent organism. Only one parent is involved. A single organism can reproduce on its own without the need of a partner. This makes asexual reproduction efficient in terms of energy and time, as it eliminates the need for finding a mate and the complexities of mating behavior. The process occurs in very less time. Asexual reproduction is generally faster than sexual reproduction. Since it involves simpler cellular processes and fewer stages, organisms can quickly produce large numbers of offspring in a relatively short period. This is advantageous in stable environment where rapid population growth is beneficial. The offspring produced are exact copies of the parent. There is no variation. The offspring are genetically identical to the parent, also known as clones. This lack of genetic variation means that the offspring inherit all the genetic traits of the parent. While this can be advantageous in a stable environment, whereas the parent's traits are well suited for survival, it can also be a disadvantage in changing environments where genetic diversity is beneficial for adaptation. The growth of the offspring is rapid. Since the offsprings are genetically identical to the parents and are often produced through efficient cellular mechanisms, they can grow and mature quickly. This rapid growth can help the species quickly colonize new areas and exploit available resources. So, here are the detailed methods of asexual reproduction. Binary fission. Binary fission is a simple form of asexual reproduction where a single parent cell divides into two equal parts. It is common in prokaryotes such as bacteria and some single-celled eukaryotes like protozoans. Examples: amoeba and paramecium. Process. First of all, the parent cell replicates its DNA. The cell grows in size and the DNA molecules move to opposite ends of the cell. The cell membrane pinches inward, dividing the cytoplasm and forming two daughter cells. Examples: bacteria and amoeba. Budding process. A bud forms on the parent organism. The bud grows and develops, eventually detaching to become an independent organism. Examples: yeast and hydra. Fragmentation. Fragmentation occurs when an organism breaks into pieces, each of which can develop into a new organism. This method is common in some invertebrates and plants like algae. Example is Spirogyra. Regeneration. Regeneration is the process of replacing or restoring damaged or missing cells, tissues, organs, and even entire body parts to full function. All living organisms, including plants and animals, have some ability to regenerate as part of their natural maintenance of tissues and organs. But underline, the degree of regeneration varies from lower animals to higher animals. That could be the reason that why can't humans or other higher animals reproduce by the process of regeneration. Example is planaria and starfish. Vegetative propagation. Vegetative propagation is a form of asexual reproduction in plants where new plants grow from vegetative parts of the parent plant, such as roots, stems, or leaves. This method ensures that the offspring are genetically identical to the parent plant. It could be natural or artificial. Here are the detailed methods of vegetative reproduction. Runners. Runners are horizontal stems that grow above the ground. They have nodes where buds are formed. These buds develop into new plants when they come in contact with the soil. Example, strawberries. Rhizomes. Rhizomes are horizontal underground stems. They store nutrients and produce new shoots and roots from their nodes. This allows the plant to spread laterally and form new plants. Example, ginger. Tubers. Tubers are swollen underground stems or roots that store nutrients. They have buds or eyes that can sprout and grow into new plants. Example, potatoes. Bulbs. Bulbs are short stems with fleshy leaves or leaf bases that store food. They have a central shoot that can develop into a new plant. Example, onions. Corms. Corms are short, vertical, swollen underground stems that store nutrients. They have nodes and internodes from which new shoots and roots develop. Example, gladiolus. Suckers. Suckers are shoots that arise from the base of a plant or from its roots. These shoots can develop into new plants when separated from the parent plant. Example, banana. Banana plants produce suckers, also known as pulps, which grow from the base of the parent plant. These can be separated and planted to grow new banana plants. Offsets. Offsets are small, complete daughter plants that grow from the base of the parent plant. They can be separated and planted independently. Example, spider plants. Cutting. Cutting involves taking a piece of a plant such as a stem, leaf or root and planting it to grow a new plant. This method is widely used in horticulture for propagating plants. Example, coleus. Layering. Layering involves bending a low-growing stem to the ground and covering it with soil, allowing it to develop roots while still attached to the parent plant. Once rooted, the new plant can be separated and transplanted. Example, blackberries. Grafting and budding. Grafting involves joining a piece of stem called scion from one plant to the stem called rootstock of another plant. Budding is similar but uses a bud instead of a stem. Example, apple trees. Micropropagation. Micropropagation, also called plant tissue culture, is a method of propagating a large number of plants from a single plant in a short time under laboratory conditions. This method allows propagation of rare endangered species that may be difficult to grow under natural conditions, are economically important or are in demand as disease-free plants. To start plant tissue culture, a part of the plant such as stem, leaf, embryo, anther or seed can be used. The plant material is thoroughly sterilized using chemical treatment. Under sterile conditions, the plant material is placed on a plant tissue culture medium that contains all the minerals, vitamins, and hormones required by the plant. The plant part often gives rise to an undifferentiated mass known as callus, from which individual plantlets begin to grow after a period of time. These can be separated and are first grown under greenhouse conditions before they are moved to field conditions. Advantages of vegetative reproduction. Rapid propagation. New plants can be produced quickly and efficiently. Genetic uniformity. Offsprings are genetically identical to the parent, preserving desirable traits. Bypassing seed dormancy. Plants that have difficulty reproducing through seeds can propagate effectively. Disadvantages. Lack of genetic diversity. All offsprings are clones of the parent, which can make them vulnerable to diseases and environmental changes. Resource competition. Closely growing cloned plants may compete for the same resources, such as light, water, and nutrients. Vegetative reproduction is a vital method in agriculture and horticulture, enabling the propagation of plants with desirable traits and ensuring consistent crop production. Spore formation. Spore formation involves the production of spores, which are specialized cells that can develop into new organisms without fertilization. This method is common in fungi, algae, and some plants. Process. The parent organism produces spores through mitosis. Spores are released and dispersed by wind, water, or other means. And when conditions are favorable, spores germinate and develop into new individuals. Example, fungi like bread molds, rhizopus, and ferns. Parthenogenesis. Parthenogenesis is a form of asexual reproduction where an egg develops into an individual without being fertilized by a sperm. This method occurs in some insects, reptiles, and amphibians. Process. An unfertilized egg undergoes cell division and develops into a complete organism. For example, in honeybees, unfertilized eggs develop into male drones. So, these methods illustrate the diversity and efficiency of asexual reproduction in various organisms, allowing them to rapidly produce offsprings and adapt to their environments. Advantages of asexual reproduction. Efficiency. It allows organisms to reproduce without the need for a mate, saving time and energy. Consistency. In stable environments, it ensures that successful genetic traits are passed on without alteration. Rapid population increase. It can lead to a quick increase in population size, which is beneficial in environments where resources are abundant. Disadvantages. Lack of genetic diversity. All offsprings are clones of the parent, which means there is little to no genetic variation. This can make the population vulnerable to diseases and environmental changes. Accumulation of mutations. Since there is no genetic recombination, harmful mutations can accumulate over generations. But overall, asexual reproduction is a straightforward and efficient means of producing offsprings, ensuring the rapid growth and spread of species, particularly in stable environments where the parent's traits are advantageous.